to Introduction to the Cell. I apologize in advance, there's a really loud fan in the background for some reason today. So you might have really loud background noise, which is aggravating, I would think, but don't really have a choice, unfortunately. So, Introduction to Cells. If you remember from a couple lectures ago, we talked about what the cell actually was. In fact, one of the properties of life, all living things have cells and are made from cells. A cell was described as the fundamental unit of life. That's the less technical definition. The more technical definition I'm not going to write up here, but if you remember it, it was a membrane surrounding a cytoplasm with various organelles carrying out certain functions. Right? That was the technical definition of it. We've known about cells for a while. But when did we first discover their existence? Most of them are so small you can't see them with the naked eye, and so prior to the invention of microscopes, generally you really couldn't see individual cells. This is the gentleman who named them. His name was Robert Hooke. He lived in England. He had a microscope, obviously not quite technologically comparable to what we have today, but it gave him close enough views of cork trees that he was able to draw this sketch of cork tree cells. Looking at it from above, the individual cells to him looked like rooms. And so the word cell can also mean a room, like a jail cell, right? And so he named them cells because that's what they reminded him of. They looked like tiny little rooms. In the years since Robert Hooke's discovery, many additional discoveries have been made in regard to cells. We now have something called cell theory. A theory, if you remember scientifically, is very different from everyday usage of the word. It doesn't mean a hunch or a guess or one of several possible explanations. A scientific theory is well supported by thousands of different studies, ultimately leading us to one particular explanation for a scientific phenomenon. In this case, we're explaining several things actually about cells. So cell theory postulates, fancy word for says, that all living things are composed of cells. We established that in the very first lecture. We talked about the properties of life. All living things are composed of cells. They have not found any life form that does not contain something that at least could be. Second, cells are the fundamental unit of life. Right, that's the definition of them. Established that. Third, cells do not arise spontaneously, they only arise from other cells. In other words, organisms don't just spontaneously pop up. Organisms are produced from the union or splitting, if you're doing asexual reproduction, of other organisms. And so cells do not arise spontaneously, they only arise from other cells. As far as the origin of the first cells then, that's beyond the scope of this course and this topic in particular today. So, cells are too small to observe with the naked eye. Most of them are. Not all of them, most of them. Since Robert Hooke's initial discovery, we've had a lot of upgrades, so to speak, in the technology of observing the small. So let's look at microscopes. You guys have the microscope lab that you're doing as well, so hopefully this will help supplement that. I use the same stock image of a microscope for 3 straight slides, so I apologize for that. But this is a compound light microscope. So a microscope is an instrument that magnifies objects too small to be observed. It's right in the name, observing something small. There are 2 major types of microscopes. This is a compound light microscope. It is one of two different types of light microscope that we have. So light microscopes, as the name implies, they use visible light to illuminate an object. The two different ones of these that we have are compound light and a microscope called a dissecting scope. Both of these are light microscopes, and up until the 1950s-ish, these were the only microscopes that existed. So light microscopes are the ones that you probably think of when you think of a microscope. And they simply use visible light to illuminate an object. And again, until the mid-20th century, mid-1900s, this was the only type of microscope that existed. So let's talk a little bit more about compound light microscopes. And there's that same picture for the third straight slide. Compound light microscopes, they let us look at flat, thin organisms or cross-sections of organisms. There's a lot more to this in the microscope lab that you will be looking at. But just an overview, the slide goes here on the stage. You can observe through the objective and the ocular lens, which magnify between 40 and 400x normally, 40 and 400 times the naked eye. Light comes from below. Light comes from below and illuminates. It has to be a very thin organism, all right, or it has to be a cross-section, so you're looking at a very thin cross-sectional slice. Right? This type of microscope requires that kind of specimen. So it actually can magnify up to 1,000. We don't use it beyond 400, but technically it does have an objective lens that goes up to 1,000 times magnifying power. The image that you get ultimately is two-dimensional. And as we said, light passes through the specimen from below, and you get a two-dimensional view of whatever it is that you were looking at. I believe the next slide has some sample pictures taken under a microscope. The technical word, by the way, for pictures taken under a microscope is a micrograph. So let's take a look at some micrographs. Here is one that you guys would have looked at if we had in-person labs. This is a onion epithelial tissue, so this is the very thin skin of an onion. And this one has been stained so that you can see the nucleus much more clearly. But you can see the individual cells. These are plant cells, which means that they have cell walls, which is why you see such a rigid, organized structure here. Here's another example of an image you can get through a compound scope. This is a cross-section of a leaf. So ordinarily, right, you would look at a leaf like this. But this is looking at it basically turned to its side. Very thin. So you can see the sections there of the leaf, the mesophyll part in the middle, that's where the plant does photosynthesis. See the epidermis protecting the inner mesophyll. Don't worry about knowing these terms for this particular section. All right, you'll have to know this stuff for a later lab activity. But for now, just use it as an idea of what a compound scope can give you. Here's another cross section. This is a cross section of the intestinal tract of some small animal. So this is the equivalent of what's called a microvillus in your own digestive tract. It increases surface area to absorb stuff as it goes through your digestive system. Anyway, the point is this is a cross-section of an intestine, cross-section of a leaf, top view of some onion skin, all two-dimensional, but it gets you close enough to see individual cells, which is the coolest part, I think, of when we get to look through these. And you may have noticed that the loud fan just turned off now. So I won't have to shout quite as loudly as we do this. All right, next one, dissecting microscopes. So these also use light, but they use light from above rather than below. So our compound scopes use light coming up from beneath the organism. With our dissecting scope, The light comes from above, right? Trying to see where the light source, like right here would be the light source, I think, for this one. And it illuminates the object specimen from above. I love these dissecting microscopes because they give a three-dimensional image, which is really, really cool. It does not magnify nearly as much as the compound light. The compound light magnifies up to 1,000 times, or even just 400 is the one that we usually use in class. But the dissecting microscope magnifies only 30 times. But as I said, it's a three-dimensional image, which is really cool. And let's take a look at some sample pictures through a dissecting microscope. Here we have a yellow jacket. You can see the individual hairs. The magnifying power is strong enough to see the hairs not only on the face, but you can see them all over its legs as well. You can see the different segments of the insect as well, which is pretty cool. This is a close-up of the flower. You can see the stamens right here, which contain pollen. You can see some pollen here. You can see individual pollen grains, and that has to eventually get into the carpel and fertilize the flower. But you can see really cool structures that the naked eye just can't perceive. But as you can tell, it's not strong enough magnification to get us close enough to see most individual cells, like our compound light was able to do. Electron microscopes. Alright, so we talked about light microscopes being the only microscopes up until the 1950s or so. In the 1950s, electron microscopy was discovered. How does this work? Well, instead of using visible light to illuminate an object, electron microscopy shoots a beam of electrons at an object coated in a particular substance, and that's going to relay that image ultimately back for us to observe. But the great thing about this is that it allows us to magnify objects to an extent that visible light magnification with our standard compound and dissecting scopes just can't get us to. The two main types of electron microscope are SEM and TEM. The scanning electron microscope is my favorite. It gives us a three-dimensional image of an object, and it gives us incredible close-ups of microscopic organisms. The TEM, transmission electron microscope, gives us a two-dimensional cross-section. This one is really cool because it gets us incredibly close. We can see the very folds of the organelles within the cell. You can see the actual folds of the walls of a mitochondrion with this TEM microscope. In terms of magnifying power, the most powerful ones, to my understanding, 10 times magnification, or 10 million times, 10 million times magnifying power. So ours gets up to 400, maybe 1,000. These get up to 10 million. As you would imagine, they are extremely expensive pieces of equipment. We do not have one at ECC. I'm guessing that Missouri S&T here in Rolla probably has one. Mizzou would probably have one as well, but we do not. Here's a picture of an electron microscope. Right? It's much larger than the compound light and the dissecting scopes that we have. But you can get really, really close. This is a close-up of an individual, I believe it is a louse, that is the singular for lice, the fun stuff that gets in your hair, right? You can see the individual proboscis here. You can see the hairs on the legs. In certain species, you can see the multi-facets of the eyes. It's just really, really cool. This is a scanning. These are both with the scanning electron microscope, the SEM. This is three-dimensional imagery. These are E. coli bacteria, individual bacteria magnified to such a degree you can see the surface of them quite clearly. Images obtained using the SEM microscope are in grayscale, and because you have to coat the organisms that you're observing, I forget the substance you have to coat them with, but that plus the beam of electrons inundating them, it only works on organisms that are dead. I have this creature here as one I just want to talk about really briefly. This is called a tardigrade. The common name for them is the water bear. They are a very interesting creature in terms of their extremophiles. They can survive so this is a creature down here. So this is a small organism. It lives in freshwater environments like ponds. One time in my 6 years at ECC, we found some when observing pond organisms. So they're here. So they're around. But they can survive for a couple of minutes at 304 degrees Fahrenheit. They can survive for 30 years at -4 degrees Fahrenheit. They can survive for a few days at -328 degrees Fahrenheit and a few minutes at -458 degrees Fahrenheit. If you're curious, that's 1 degree Kelvin, that's 1 degree above absolute zero when atomic motion ceases to occur. They can also withstand the vacuum of outer space, the only organisms that, as far as we know of, can be put out in space without protection and survive. They can survive without any water, completely dry for 10 years. They can survive 1,000 times more radiation than any other animal as far as we know of. So these guys, at least in the animal kingdom, are the most extreme survivors that we have. Pretty cool creatures. All right. So water bears or tardigrades, they're pretty small, but even they are not just one cell, they're multiple cells. And if you look at single-celled creatures like the amoeba, for instance, very, very small. A single egg, as far as I know, is one of the largest cells that we have. And that's one of the few that we can actually observe. A single chicken egg, for instance, is one cell. But most cells are very, very small. And we want to go into just how small here by talking about some distance perspective. If you remember the metric staircase, we had kilo, hecto, deca, and then we had the base, which was the meter, right? Base here, so don't get it confusing. Base, deci, centi, milli. Now I want you to think, okay, a meter is about this long, roughly, right? Roughly that long. And a millimeter is the size of a little pinhead, if even that. So going from a millimeter, 3 steps up the staircase takes us from the size of a little pin to this. Now, going down the staircase, 3 more, we get to the level of micro. So, meter, millimeter, micrometer. 1/1000th of a millimeter. In other words, if you blew up in size everything around you so that a millimeter was now this size, a micrometer would then be the size of a millimeter, the size of a little tip of a pin. So we're talking small. And most cells are between 10 to 100 of those little micrometers in size. Which begs the question, why are cells so dang tiny? It's a great question to ask. Whenever I ask it in class, nobody gets the answer, the correct answer. Here is the current accepted explanation for why cells are so small. Here is a hypothetical larger cell. we're going to say the volume of this hypothetical cell is 100 cubic micrometers. That's how much space it occupies. Here's a bunch of smaller cells that take up the same amount of space when put together. The volume is the same. The amount of space they occupy is identical. But what does this side have a lot more of than this side? The answer is surface area. By having so many smaller cells, you've got a lot more cellular surface area exposed than you would have if you had one giant cell, or even just a large number of bigger cells than you have. Smaller is better for maximizing surface area. Why is that important? Well, a lot is going on at the cell's outer membrane. You've got transport proteins, you've got receptor proteins, you've got the movement of ions and water and glycoproteins communicating between the cell and the outside world. There's just a whole bunch of stuff going on there. You want to maximize the amount of surface area with your cells. If you make it smaller, there's more surface area. Imagine if you cut each of these into 9 pieces, you'd have even more. A good way to think about it is if you like to cook chicken, if you've got one giant slab of chicken, right? I'm not even going to try to draw it in the shape of a chicken. If you've got one giant slab of chicken and you try and cook that, it's going to take a lot longer to cook than if you chop it up into small pieces. Because when you chop it up into small pieces, it may be the same amount of chicken that you're cooking, but there's so much more exposure to the heat, surface area, and so you're going to cook it a lot more quickly now because you've got so much more exposure of surface area. In this case, it's to the heat of the frying pan, but in your cell's case, it's just more surface area means more space for cellular work to be done. So cells are small because being small gives them a higher amount of surface area relative to their total volume. As we said, the surface of the cell is a busy place with water, ions, and other molecules constantly moving in and out. And being small maximizes available surface area. Awesome. All right. See if you guys remember these words. Hopefully you do. Eukaryote versus prokaryote. You are a eukaryote. That means your cell has a nucleus. So if this was your cell, there is a nucleus. That's a eukaryotic cell. Prokaryotes don't have a nucleus. That's their main defining feature. It's what they don't have. And so prokaryotes, or prokaryotic cells, there are 2 types of them. If you remember back, one familiar and one much less familiar. The familiar one is the bacteria. So, prokaryotes, cells without nuclei. The familiar one is the bacteria. The less familiar one is archaea. If you remember them, they're the ones that live in the extreme environments, like geysers and deep sea vents and such. So, main defining feature of prokaryotes do not have a nucleus. I forget exactly what bacterial species this was, but this was taken with a SEM, scanning electron microscope. If you take microbiology, these guys will have their 15 minutes of fame. We're not going to talk about them too much, right? Because we're focused more on eukaryotes in this class. But we're going to look at a couple of features that all prokaryotes tend to share, or at least a large number of them do. Specifically, we're talking about bacteria when we do this. Archaea, they're an interesting group, but we don't talk much about them at all. So here are parts of a typical bacterial cell illustrated using a beautiful Microsoft Paint picture. First thing to remember is that bacteria, they don't have a nucleus because they're prokaryotes, but they still have DNA. But the DNA is not randomly strewn about. It exists in the middle region of a bacterial cell. So this is the bacterial cell. This purple strand here is DNA. A lot of bacteria have a singular strand of DNA called a plasmid, but they have DNA, and it's in that central region. I want you to look at the second word up there. It's nucleoid. Think of the word, or the suffix, oid. Think, this is the best example I have for this. Think about an alien that is a humanoid alien, right? You can tell they're kind of human-like. Maybe they have 2 arms, 2 legs. They look kind of human-like, but they're not really human. Nucleoid region in bacteria, it's like the nucleus of your cell, but it's not really a nucleus, just like humanoids aren't truly human. So this is the nucleoid region of the bacterial cell. It's where the DNA is. It's not in the nucleus, but it's in a nucleus-like place. We'll talk about ribosomes later. Don't worry about them for now. The cytoplasmic fluid is the fluid within the cell membrane. It contains all of the organelles within. Your cells have a cytoplasm as well. The plasma membrane, that surrounds every single cell, including all of yours. Some bacterial cells have strong cell walls. Some of them have sticky capsules. Some of them have a whip-like tail called a flagellum. Plurally, it's called a flagella. Or they're called the flagella if it's plural. But these are features often observed in bacterial cells. Like I said, we don't talk about them too much in here. Microbiology goes into much more detail about the life history of these guys. We are interested in eukaryotic cells. Like, we're interested in all of them, but we're going to focus on eukaryotic cells. So these have the true nucleus, right? This is animals and plants and fungi, and then those protists like amoebas. We're going to look at some differences between animal and plant cells in this unit, because there's a lot of similarities, but there are also stark noticeable differences that we want to get into. And these 2 wonderful pictures are also drawn in Microsoft Paint. The one on the right is a plant cell. You can tell that from the presence of the cell wall, among other things like chloroplasts and this giant thing, which we'll talk about later. But this is an animal cell, this is a plant cell, and the next unit will look at each of the organelles within the cell and how they help the cell carry out all of its functions.